This is Solomon, and I'm going to be talking about histology today. Histology is the study of tissues, and a tissue is a group of similar cells organized into a structure and functional unit. In other words, it's a group of cells that have the same structure that are organized to carry out a particular function or to carry out particular functions. Now, tissues must be physically linked. They might have intercellular substances or they might not. And also, they may comprise one or more type of cells. But the purpose is they should be specialized to perform a particular function. So that should be important as we go on. Let's talk about why we need to study histology. First of all, we need the study of histology in hospitals. When we are checking out what is paining you, what disease is disturbing you, we check blood samples. And it's also important to understand that cells do not behave or do not work in isolation. So we need to study histology to be equipped for such a day when we need to analyze different types of tissues. So it's important to understand that both plants and animals have tissues. Let's talk about animal tissues. Now, early in development, the cells of the growing embryo differentiate into three germ layers. And then that's the ectoderm, the mesoderm, and the endoderm. The ectoderm forms the outer layer of the skin and the nervous system. The mesoderm forms muscles and connective tissues, skeleton, kidneys, circulatory system, and reproductive organs. And the endoderm forms the lining of the respiratory tract and the urinary bladder. It also forms the glands associated with the guts and the respiratory tract. Now, therefore, the germ layers, the ectoderm, the mesoderm, and the endoderm, differentiate into different cell types and tissues that are characteristic of the vertebrate's body. Tissues are joined to each other by proteins. The point of connection between two cells is what we call a junction. So, for example, in our plants, we have what we call the junctions in between, and those holes, those junctions, are going to be called plasmodesmata. And then we are going to have the gap junctions. We are going to see them. In adult vertebrates, there are about 5 types of tissues: the epithelial tissue, the connective tissue, the muscular tissue, the nervous tissue, and the reproductive tissue. And these eventually connect together to form the organs of those particular areas. For example, the reproductive tissue eventually form the reproductive organs, and so on and so forth. Let's talk about epithelial tissue. Epithelial tissue, a collection of closely packed single and multi-layered compound sheets of cells. So these ones line cavities, surfaces of blood vessels, and organs. They can be simple epithelial tissue or they can be compound epithelial tissue. We basically have 3 shapes. They are either squamous, that means they are flattened, they are columnar, or they are cuboidal. And epithelial tissue do not have blood vessels. They receive their nourishment simply by diffusion, because usually they are either one or just a few cells thick. They are all attached to a basement membrane. And the portion of the epithelial cells that is attached to the basement membrane is called the basal surface. And the opposite end, facing the external environment, is what we call the apical, or what we call the free surface. They have a high regeneration capacity. They are constantly dividing to replace the ones that have been worn out, for example, under an injury or in case of abrasion or friction, and so on and so forth. We are going to discover that they are found around all surfaces. We have those epithelial tissue. What are their functions? They serve the function of secretion, selective absorption, secretion, because we are going to see that there are some epithelial tissue that are glands, and then selective absorption, we are going to find them in the ileum. Protection, we are going to find some of them on the skin, the vaginal tract, transcellular transport, and then sensitivity. Because they are found lining cavities, it makes sense that they should also be modified for sensitivity. So those are the major 5 functions of epithelial tissues. Let's talk about the simple epithelium. Under the simple epithelium, we have what we call the simple squamous epithelium. This can also be called the pavement epithelium. It consists of a single layer of cells. These are so thin that the nucleus actually causes a bulge. Okay, the nucleus bulges out and the nucleus is centrally placed. They form a disc shape. They are irregular and they have tapering edges. The simple squamous epithelium are found in the following areas. They are found in the renal corpuscle, what we are calling the Bowman's capsule. They are found in capillaries. They are found lining the alveoli of lungs, lining the blood vessels. And when they are lining the blood vessels, they are called the endothelium. The blood capillary walls and linings of the lymphatic vessels. This is an example. That's a picture of the simple squamous epithelium. So we have the basement membrane, just before, and then we have the simple squamous cells. So you can see the nucleus in the center of irregular cells, and then just below they are attached to the connective tissue. So if this was an artery, this would be, for example, the smooth muscle or the elastic, the smooth muscle that then is attached to the endothelium. Their function, majorly, their function are diffusion of materials and exchange of gases. Maybe then we can ask the question, how are they actually adapted to perform this function? Now, the adaptation of the epithelial tissue is that they are thin and they are flattened. Them being thin and flattened serves to reduce the diffusion distance of materials. And then they possess smooth linings, which allows relatively friction-free passage of fluids and materials. That is why they are very important to be found in the red blood cells, in the arteries and the veins and in the capillaries. Why? Because they have a smooth lining which allows materials to move without friction. The simple cuboidal epithelium. These cells are roughly cube-shaped. They possess a central spherical nucleus. The upper surface of cuboidal cells can either be pentagonal or it can be hexagonal in outline. And then they are the least specialized of all the epithelial tissue. So you can talk about them being attached to the basement membrane. They have a nucleus at the center. Their function is majorly secretion, and they line organs. They are found in the salivary ducts, in the sweat glands, in the thyroid glands, the pancreatic ducts, the convoluted tubules of the kidneys, the collecting ducts of the nephron, the salivary glands, the sweat glands, the thyroid glands, and they're even found lining the retina. So that's if you were to be asked where do we find the cuboid epithelium, you can just go back and watch that previous section. What are their function? Their function is protection. Protection, because they are found in the convoluted tubules, we can also say they are found in excretion, absorption, and they also have the function of secretion. It's important to note that the simple cuboid epithelial tissue is non-secretory in the proximal convoluted tubule and the distal convoluted tubule and the pancreatic ducts. In those areas, they do not perform the function of secretion. They perform other functions like excretion and absorption, but they do not perform the function of secretion. How are they adapted? They have cells that are tightly packed. As you can see, these cells are tightly packed here, and they have very little intercellular spaces, which make them able to offer protection from injury and refuse pathogens from accessing other parts of the organ, which offers protection from infection. They have very many Golgi bodies, which perform functions of secretion of hormones and enzyme, and some possess microvilli, which then help them to increase the surface area, for example, the reabsorption of materials in the kidney tubules. Some of these have numerous mitochondria for energy production, which can then be used for active reabsorption of materials from the renal filtrate back into the bloodstream. The simple columnar epithelium. These ones are tall, narrow cells. If you're observing them at the nucleus, they are very tall cells, and their nucleus is found sometimes at the bottom. Okay, so each cell possesses a nucleus that is situated at the basal end and it's oval in shape. The epithelium is often interspersed with goblet cells, as we are going to see. The free surface of each columnar cell in most cases has microvilli that form a brush border. Where do we find them? We can find them in the digestive system, in the small intestines, in the gallbladder, in the kidney ducts. They are found lining gastric glands, the intestinal glands, the mammary glands, the thyroid glands, the salivary glands. All those glands, we are going to see later even that they have the simple columnar epithelium. Now, from where they are found, we can actually derive their function. For example, because they are found in glands, we can say one of their functions is secretion. Because they are found in the intestines, in the stomach, we can say one of their functions is protection, absorption, and the brush border they have increases their surface area by having microvilli at the cell-free surface. How are they adapted to their function? They possess finger-like projections that we call microvilli, which increase the surface area for absorption, such as digested food in the intestines. Another adaptation that we can talk about is them having a mucus-secreting cells which secrete mucus. The mucus protects the gastric walls from hydrochloric acid and the digestive enzymes. And then finally, the mucus from goblet cells also lubricates the passage of food in the intestines. Let's talk about ciliated epithelium. Cells of this tissue are usually columnar in shape, but they bear numerous cilia at their free surface. The cells are usually associated with mucus-secreting goblet cells. So you can see the mucus in this picture that we have here, and then you can see the cilia, and each of these is one cell, so we call it a ciliated cell. This small cell here is going to be the replacement cell for the goblet cell when it eventually gets old or weared out. Where are they found? These ciliated epithelium can be found on the linings of the oviduct, the ventricles of the brain, in the spinal canal, in the respiratory passages, for example, the trachea, the bronchi, and the bronchioles. What are their functions? They produce mucus, as you've already seen, which protects the lining and lubricates the passage of materials. The cilia, they set up currents to move materials in certain direction. For example, in the trachea, where they keep on beating upwards such that any dust is going to be collected in mucus and eventually is going to be pushed out of the tracheal tract. How are they adapted to their function? They are interspersed with goblet cells. As you can see the goblet cell in that image there. They are interspersed with goblet cells which secrete mucus to protect the lining of the gut from the enzyme and acidic action. They possess cilia which set up currents that move materials from one direction to another. They possess goblet cells which lubricate the passage, because remember, the goblet cells, they produce mucus. The pseudostratified epithelium. Let's talk about those. Now, the word pseudo means false. So when you hear pseudostratified, means they appear stratified, but they are not actually stratified. So this is a simple epithelium, since all cells in this epithelium particularly rest on the basement membrane. If all cells were not resting on the basement membrane, then it would be stratified. So you can see this cell here runs up to down here. This one runs up to down here. This one comes back to the basement membrane. All the cells in this particular epithelium are found, they start from the basement membrane. This gives an appearance. So the problem is not all of them reach the free surface. So when you look at it, it gives an appearance of an epithelium being on different levels and the nuclei having different layers. But in actual sense, all the cells actually get back and reach the basement membrane. Nevertheless, the epithelium is one layer of cells which is attached, all of them attached to the basement membrane. Most of the cells in this particular epithelium are columnar, thus they are called the pseudostratified columnar epithelium. When they have cilia, they are going to be called pseudostratified columnar ciliated epithelium. For example, they are found in the trachea, in the bronchi, and in the bronchioles. Pseudostratified epithelium also lines the urinary tract. This type of epithelium is also found in the urinary tract, and it is also found in ducts of large glands where it is not going to be ciliated. So let's talk about the stratified epithelium. Now, these are what we call the compound epithelial tissue. In the simple epithelial tissue, all cells are attached to the epithelium. But when it comes to compound epithelial tissue, we are going to discover that not all cells will be attached to the compound, to the basement membrane. So let's start with stratified epithelium. It is made up of many layers of cells with only the bottom layer resting on the basement membrane. So you can see here that only this layer of cells actually has access to the basement membrane. The rest are lying on top of each other. And as we go up, they become more squamous. So these cells, the cells that are attached to the basement membrane, form what we call the germinative layer. And these ones are continuously dividing by mitosis. And the more they divide, they push others on top. They push other areas outwards. They are primarily found in areas where the epithelium plays a role of protection. While the other simple epithelium had the role of secretion, absorption, exchange of materials, these ones in particular are found in areas where the epithelium has the role of protection. So we have what we call the stratified squamous. So in the stratified squamous, the cells first formed on the basement are cuboidal in shape, but as they are pushed outwards to the free surface, they become flattened and they become squamous. It is the thickest of all epithelia, and its function is purely protection. The cells of the surface layer may even be keratinized, or what we call conified, or lack keratin. Now, when they lack keratin, they are called unconified, and when they have keratin, they are called conified. Keratin is a tough protective protein which prevents water loss and is resistant to friction and even repels bacteria. Non-keratinized stratified squamous epithelium are found lining wet surfaces, and they are subjected to abrasions such as the lining of the mouth. For example, in the esophagus, in the epiglottis, in the vagina, all these ones here are non-keratinized. However, the keratinized version is found on the epidermis of the skin. So as old cells are removed, new cells form and the old ones are pushed up. The cells near the surface are flattened, and as time goes on, every time, the cells on top will always be renewed. Let's talk about transition epithelium. Transition epithelium is a modified form of stratified epithelium. It has about 3 to 4 layers of cells, and all of them are of similar size and shape, apart from at the free surface where they become more flattened. These cells do not break off. It is found in organs which can expand, for example, the urinary bladder. And in the women, the girdles of women which come back to normal after delivery. It is found in organs or structures which can expand, like the urinary bladder or the urinary tract, the ureter, the urethra, the bladder. So here, the cells of the basal layer are cuboidal and columnar, while the cells of the superficial layer, the layer on top, they vary depending on how distanced the tissue is. In, for example, urinary bladder, when it is not distanced, the top layers appear cuboidal. But when it is stretched, they appear squamous. And these ones do not have a basement membrane. So because the shape of the cells is transitionary, that means it changes depending on how much stretching the organ has undergone. The epithelium is called transition. How is it adapted? The transition epithelium is able to change its shape, which allows the organ to be able to expand. For example, in the urinary bladder, which can increase its volume. The transition epithelium is also composed of very many layers of cells, which makes it largely impermeable to water and blood and urine. And because it is thick, urine is unable to escape from its surrounding tissues. So when we come next time, we are going to be talking about glandular epithelium. So please subscribe and like and turn on the notification bell if you're interested in watching more videos like this.